Welcome to Cell Therapy Narrative. I'm a PhD biomedical research scientist specializing in cell therapies. Today we're going to discuss the review paper titled From Concept to Cure, the Evolution of CAR T-Cell Therapy. The corresponding author is Carl June, who is an MD and specializes in oncology, and he is one of the pioneers of CAR T-cell therapy. This paper was published in the journal Molecular Therapy in May of 2025. The authors to this paper do an excellent job of telling the story of how CAR T-cell was developed and how it became a groundbreaking therapy which emerged from decades of iterative advancements. As of February 2026, the FDA has approved 7 CAR T-cell therapies, all second-generation designs, marking a major milestone in engineered cell therapies for blood cancer. What is CAR T-cell? Let's first talk about what T-cells are. T-cells are a type of white blood cell called lymphocyte, which help to fight infection. There are 2 types, the cytotoxic T-cells that kill infected cells, and the second type is the helper T-cell that sends signals that direct other immune cells to help fight the infection. CAR T-cells therapies use bulk T-cells, meaning they use a mix of both types. Now, CAR stands for chimeric antigen receptor, part antigen and part receptor. Scientists took the T-cell receptor and engineered it to contain an antigen binding domain. This part recognizes the cancer cell and will bind to it. The other part of the receptor is the co-stimulatory domain, which activates the T-cell. Now, how do they get that receptor onto the T-cell? They use a virus that they engineer to contain the genetic material that codes for that specific CAR, and they introduce the virus to the cell. So the virus enters the cell and integrates this genetic material into the T-cell genome. The T-cell will use its transcriptional machinery to sit down on the DNA and read this code or transcribe this message, and then it will translate the message and make the receptor. That receptor will locate to the cell membrane of the T-cell. And now this T-cell is known as a CAR T-cell and can recognize and bind to the cancer cell and kill it. How does CAR T-cell therapy work? First, they take blood from the patient and isolate the T-cells. These T-cells are put into a dish and they do all of their manipulation in this dish. Next, they use a viral vector to genetically modify the T-cell's genome to have the CAR encoding gene. The T-cells now express CAR on their surfaces and are known as CAR T-cells. CAR T-cells are expanded out to the millions. Finally, the CAR T-cells are placed back into the patient's bloodstream. These CAR T-cells can now recognize the cancer cells and will kill them. This technology is really taking off. There has been success for blood cancers and there's active research for other major conditions. Now that I've introduced CAR T-cell therapy, let's go back and start from the beginning to learn how it was developed. The first question we're going to ask is, how did early scientific studies lead to discovering a correlation between the immune system and cancer? We're going to look at a famous experiment and story that occurred in 1796. You may have heard of this one. An English country physician by the name of Dr. Edward Jenner first observed that milkmaids who contracted cowpox disease were immune to smallpox. If you remember, smallpox was a deadly disease killing 1 in 3 people. Dr. Jenner took pus from a cowpox blister from the hand of a milkmaid. He then inoculated an 8-year-old boy with it. The boy developed a mild fever and a blister, but quickly recovered. A few months later, Dr. Jenner inoculated the boy with matter from a smallpox lesion, and the boy showed no signs of the disease. Jenner later repeated the experiment on other children, including his own son. This led to the world's first vaccine to smallpox. This shows that taking a similar virus that is weaker trains the immune system to recognize the more dangerous virus and neutralize it before it causes symptoms and disease. He published a paper on this in 1798. In 1866, Dr. Wilhelm Busch had a patient with an advanced malignant tumor. During treatment, the patient developed a severe bacterial skin infection known as ursilopolis. To Busch's amazement, the tumor began to shrink rapidly during the fever and inflammation caused by the infection. So this leads us to ask, was the infection somehow mobilizing the body's own defenses to attack the tumor? In 1880, a surgeon, Dr. Frederick Feligin, built on Busch's finding and intentionally infected cancer patients with live cultures of the ursipolis bacteria. He observed the same phenomenon. When the body mounted a strong immune response against the infection, tumors shrank or disappeared. Now, the ursipolis bacteria is a fast-spreading bacteria, and it's quite dangerous. So The problem with this method is that the patient can develop sepsis and die. Now, in 1891, Dr. William Cawley made the treatment safer by switching from live bacteria to a killed bacteria mixture. He called the preparation Cawley's toxins. Between 1890s and the 1930s, over 1,000 cancer patients were treated with Cawley's toxins. Many experienced dramatic tumor regression and some achieved long-term remission. These studies demonstrate that the immune system can be harnessed to destroy cancer. Through animal studies, scientists determined that T cells were responsible for eliminating cancer cells. The next question we're going to ask is, what is the mechanism for T cells recognizing and killing cancer cells? Here we have a T cell and an antigen-presenting cell binding to one another. Let's zoom in on this part. First, antigen-presenting cells will engulf a stressed or dying cancer cell, break down the cancer proteins into peptides, and then present the peptides, aka antigen, on the receptor of the cell membrane. This receptor is specifically called a major histocompatibility complex. Now, in humans, this is also called human leukocyte antigen, but I'm just going to keep referring to it as MHC. The T cell receptor on the T cells combine to the major histocompatibility complex and the antigen if they match, just like a lock and key. When the T cell receptor matches both the major histocompatibility complex and the antigen, the T cell becomes activated. The T cell then undergoes cell division and expands out into lots of T cells. These activated T cells are now capable of recognizing and binding to the cancer cells and killing them. It is important to note that T cell activation requires multiple signals. Signal 1 is called CD3. It's another receptor that is in close proximity to the T cell receptor. Signal 2 is CD28. There's two different types here. And finally, signal 3, which is not in the picture, is IL-2 signaling. Now, what does activation mean? Activation means that there's a cascade of events that turn on genes that will make proteins and carry out a function. So here we have the antigen-presenting cell. It binds with the T cell receptor. This activates a downstream signaling protein, which can activate another downstream signaling protein that then translocates to the nucleus and binds to a complex of other proteins at the part where they want to read the DNA message. So these are called transcription factors. They read or they transcribe the message. And this turns gene expression on. So now it will make the protein, which will have a function. And so different receptors activate different genes, and this carries out a specific function. All right, so now how and why does cancer occur? So one reason is that you have no T cells that are capable of recognizing their tumor antigen. The second reason is that we do have tumor-specific T cells, but they aren't sufficiently activated and expanded. This could be because cancers are our own cells and are considered self, and our T cells were trained in the thymus to not react too strongly to self-antigens. Therefore, they might not bind too strongly to the antigen and its MHC. This prevents the T cells from reaching the numbers necessary for eliminating cancer cells. All right, so we've first talked about the history or the evidence that the immune system can be harnessed to destroy cancer. Then we talked about the mechanism to how immune cells, or more specifically T cells, recognize and kill cancer cells. Then we talked about why cancer occurs, or how this mechanism does not work properly and that leads to cancer. Now we're going to get into the immunotherapy treatment options. So first we're going to talk about T cell receptor therapy, how that works, who it's for, and what the problems are. And then finally, we're going to get into the groundbreaking CAR T cell therapy and talk about how that overcomes the challenges associated with T cell receptor therapy and how it's really taking off as treatment options, not just for cancer, but for other diseases as well. First, we're going to talk about the immunotherapy called T cell receptor therapy. Scientists engineered the T cell receptor to better bind to the tumor antigen. This is different than CAR T cell therapy, which we will get to soon. So this image looks really similar. This is the same slide that I used for the CAR T cell therapy introduction. But we're going to be talking about T cell receptor therapy, which is very similar and works pretty much the same way. So you take the patient's blood, isolate the T cells. You use a virus to genetically modify the T cell genome that will code for a new type of T cell receptor. This then gets expressed in the cell membrane of the T cell, expands out to the millions, and then gets put back into the patient's blood and can fight the cancer. Now, T cell receptor therapy only works in 10 to 20% of patients. The T cell receptor can be modified to match the cancer antigen, but the same is not true for the MHC. Everyone has their own unique signature for the MHC. If you remember, I said that the T cell receptor needs to be a match for both the cancer antigen and the MHC. And in this case, the T cell receptor is only modified for the cancer antigen. So therefore, if people have their own unique signature for the MHC, then it might not bind too strongly to the combination. Therefore, the modified T cell receptor is only going to be a match for a certain percentage of people. The second problem is that tumors can shut down the MHC completely. Therefore, it's no longer present on the cell membrane. Therefore, the T cell receptor will not be able to bind to the antigen-presenting cells without it. All right, so now we can get into CAR T cell therapy. CAR is a synthetic engineered T cell receptor that does not need to bind to the MHC, so it's MHC independent, which is a good thing because that solves the problem that everyone has a unique MHC signature. Now you don't have to worry about binding to it at all. Now, the second part of CAR is that there's no need for the activation of the other signals because it is already built into the receptor, so it will automatically become activated. Therefore, this eliminates the need for the T cell to be activated by the antigen presenting cell. The modified T cell with CAR will go directly to the cancer cell. So there has been several generations of CARs, and we're going to go walk through them. First is generation 1. It's very simple. You have the antigen binding domain, which is outside of the cell. This part recognizes and binds to the antigen on the cancer cell, and it's been modified to specifically bind to a specific antigen. You have the hinge or spacer that goes through the cell membrane. And then finally, on the inside of the cell, you have the co-stimulatory domains. And here we have the CD3 domain. So if you remember, that is signal 1 that needs to activate the T cell. Now, the first generation of CARs were too weak to work clinically. They did not make it to clinical trials, and that makes sense because I've described to you that there are several signals that are needed to activate the T cell. So this one did not work out. So then they developed generation 2 CAR T cells, and here we have 2 separate CARs. So these are independent of each other and were tested independently. So this time they added on CD28, and you remember that's our second signal. And then in the other one, we have a different one called 41BB. Now, I didn't talk about that one before, but that's a second type of signal that can be used. Now, these second signal co-stimulatory domains, they dramatically improve expansion, survival, and cytokine signaling, and was able to induce complete remission in blood cancers. This was the first breakthrough with CAR T cell therapy, and the paper stated that each has its own advantages and both have similar efficacies. Then they go on to develop generation 3 CAR T cells. So now they have combined these 2 co-stimulatory domains together. They hypothesized that it would exhibit the combined effects of the 2. It had a stronger cytokine production and good expansion. However, this had a higher toxicity and no consistent improvement over second generation. Here we have generation 4. They focused on CD3 and CD28. They did not include 41BB. And here they added nuclear factor of activated T cells known as NFAT. NFAT is a transcription factor that activates the cytokine release system. This improves killing inside the immunosuppressive tumor microenvironment, and this may be useful for solid tumors. And our last generation that we have is generation 5. They're still using CD28 and CD3, and this time they use the cytokine receptor IL-2. So you remember I said there was 3 signals. IL-2 was the third signal, and this triggers JAK-STAT signaling, which helps CAR T cells avoid terminal differentiation and exhaustion. And this leads to better persistent and more effective long-term tumor control. Now here's a summary of the overall effectiveness of these different CARs. So generation 1 was very low, didn't make it past animal studies. Generation 2 has high effectiveness. This is the most successful. All of our FDA-approved CAR T cell therapies are approved using second generation CAR T cells. Generation 3, strong in theory, mixed in practice. It was limited, no approvals. Generation 4, now this is, they're still doing active research. It's strong and solid tumor model, so it's in early trials, and this is at the experimental stage. And the last one, generation 5, it's promising, has safer signaling, and this is also in early trials and is considered experimental. Now I want to get into talking about a couple stories of the first people to receive second-generation CAR T-cell therapy for relapsed and refractory blood cancers. The first patient to be treated was William Ludwig, very first adult to receive CD19 CAR T therapy at the University of Pennsylvania in 2010. He had treatment-resistant chronic lymphocytic leukemia, and after receiving CAR T cells, his leukemia vanished and stayed gone for more than a decade. Now, unfortunately, he has died in 2021 from complications of COVID, and he was 75 years old. The next was Emily Whitehead, the first pediatric patient treated in April 2012. She was 6 years old. She had multiple relapses in her cancer and failed the standard treatment. She received CD19 CAR T cells. Emily went on to achieve complete remission and has remained cancer-free for over 10 years. She is still alive and is cancer-free and is now in her 20s. These two successes have led to larger, multi-centered trials. Now, these early clinical trial outcomes, there was an overall response rate of 44 to 91% in B-cell lymphoma and CLL, and a complete response rate of 28 to 68%. And in acute leukemia, there was complete remission that exceeded 80%. These clinical trials led to major FDA approvals. Here I'm showing the summary of the 7 FDA-approved CAR T-cell therapies again. Now you might understand this a little bit better. You see that most of them have the CD19 target antigen, and then there's a couple BCMAs. So these are antigens that are found on the blood cancers. Then over to the right, we see the co-stimulatory domains. So they mostly use 4-1BB, but they also have a couple CD28. The first approval year was in 2017, and the most recent was in 2024. All research is being done to develop CARs for solid tumors. There's a couple challenges when it comes to solid tumors. One, they are heterogeneous, meaning that different regions of the tumor often display different antigens. So a single CAR target may only hit part of the cancer. Two, there's also this dense physical barrier made of stromal cells and extracellular matrix. That makes it difficult for the engineered T cells to reach the tumor core. Thirdly, solid tumors produce immunosuppressive signals and can exhaust the CAR T cells. Despite these challenges, there is still good progress being made to tackle solid tumors. Here are some examples of CAR T cells that are being developed and having promising effects in early clinical trials. So a lot of them here are for glioma and glioblastoma, and then we have a couple below at the bottom for GI tumors. So there is ongoing active CAR T cell therapy research. So the one that was worked on first were for blood cancers. So we have had a lot of success in treating blood cancers now. I've just talked about the solid tumors. They are working on glioblastoma, pancreatic cancer, ovarian cancer, breast cancer, lung cancer, and more. You got autoimmune diseases. They're working towards lupus, multiple sclerosis, rheumatoid arthritis, Graves' disease, and type 1 diabetes. They're working on neurodegenerative diseases, so Alzheimer's, Parkinson's, ALS, infectious diseases, such as HIV, hepatitis B, and other viral infections. And this last group that they have here is fibrotic, transplant, and other conditions. So they're working on cardiac fibrosis, liver fibrosis, graft versus host disease, and organ transplant rejection. There's a lot of exciting research being done here and a lot of good that's going to come from CAR technology. Next generation CAR T-cell therapies are being designed to make these cell-based medicines stronger, smarter, and safer. Researchers are creating CAR T-cells that can recognize multiple targets at once for more complex cancers. Finally, by using tools from synthetic biology, CAR T cells can be programmed with on-off switches and safety controls, allowing doctors to fine-tune when the cells activate, how long they last, and when they shut down. When we get that far in treatments, that will be some truly impressive biology. Thank you for listening. I hope you learned something from this review paper. This is my first YouTube video, so please feel free to leave me feedback. And also, if you have questions, leave them in the comment section below. Please like and subscribe to the channel. That would really help me get started. Thanks, guys. See you in the next one.